Great. Thanks, Claire. So welcome, everyone. I'm Mala Murthy, for those who don't know me, and I want to welcome you to our first Flywire Community Town Hall. This is for members of the Flywire community, and we're happy to give you a bit of an update today. So I'll provide kind of a state of the project update, and then I'll turn it over to Claire McKellar, who's our community manager, and Sven Dorkenwald, a computer science graduate student here at Princeton and one of the co-leads of the Flywire project. So for those who are new to Flywire, and there are many of you out there, I wanted to first bring everyone kind of up to speed on the resource. So Sven, do you mind advancing to the next slide? Great, thanks. So as you know, Flywire provides a whole brain connectomic resource for those interested in Drosophila neuroscience. And importantly, it's a community-driven resource. So it's built from data and tools that were developed by members of the community and shared openly for the benefit of science. It uses FAF-B data, which was collected at Janelia and Dobby Bach's lab, and that's shown at the left in gray. Dobby shared the data set and made it accessible to anyone, which enabled us roughly 2 years ago to launch Flywire. And our project is funded primarily through the NIH Brain Initiative. So we started by providing high quality neural reconstructions using the latest advances in artificial intelligence. And this we accomplished through realigning and then segmenting this FAF-B data set, which you can see in color overlaid on the data at the left. So our segmentation, as those who've worked in Flywire know, is good. And you can kind of see that in the middle example, comparing automated segmentation of olfactory projection neurons with proofread ones. But it's not perfect. And so it requires some manual proofreading. And this is going to be true and has been true and will continue to be true for any Connectomics data set. So we therefore built the infrastructure for facile proofreading in NeuroGlancer, which is another community resource provided by Google. Flywire also leverages synapse and neurotransmitter information that come from, in particular, Jan Funke and Greg Jeffress's labs working together between Janelia and Cambridge. So one of the updates we wanted to bring to you today is that together, through this collective effort, this resource has the potential for generating a complete whole brain connectome of a female brain within the timeline of roughly a year. And that's based on accelerating proofreading, which we'll talk about in a minute. Now, an intermediate milestone would be the completion of the connectome of the central brain by roughly April of 2021, accompanied by its public release. And, to me, this would be a significant achievement for the Drosophila community working together in this data set. As someone who's worked in the community for over 2 decades, it would give me great pleasure to see this achievement pulled off as an open science initiative fueled by Fly Labs working together. And I do want to emphasize that it relies on collaborative work. We're well aware of that. We're excited by how many labs who have joined, but I do want to say a few words on our principles of openness before I hand off to Claire and Sven. So Sven, do you mind going to the next slide? So the principles of openness for Flywire kind of grew from Sebastian's first open connectomics collaboration, iWire, which is shown in the background on the left, with one important difference, and that's that we are crowdsourcing proofreading of this data set to the Drosophila community to leverage their collective expertise of circuits throughout the brain. And of course, as the Drosophila community knows the value of connectomics, and so there are a lot of labs that are eager to begin to proofread and use this data set. So as I mentioned earlier, we're very grateful that we have over 40 Drosophila labs participating, and that number continues to grow. And these labs are actively proofreading in Flywire. I did want to mention that open science in connectomics is particularly valuable. So an example is illustrated here on the right. Imagine you're working on these neurons in red, and you proofread them in Flywire and you study their connectivity. It clearly benefits you to know about the connections with all of the neurons in blue that someone else in the community may have proofread that are clearly relevant for your circuit of interest. And so, as a Drosophila community, we have a history of open science, but it's particularly critical for connectomics. We think that as connectomics data sets come online for even larger brains than the fly, I hope our community can kind of stand as an example for how to do successful open science going forward. Sven, can you go to the next slide? So in addition to Sebastian and I, Flywire is governed by an advisory board that's been generously donating their time to provide feedback. These folks are shown here: Davi Bach, Stephanie Hampel, Greg Jeffress, Andrew Seeds, and Rachel Wilson. And going forward, the board will also play a role in resolving any conflicts that arise with data sharing or publications. And I'm happy to talk more about that in the discussion period. So with that, I want to turn it over to Claire McKellar, who's going to introduce one of the members of our advisory board, Andrew Seeds, to share a bit about the science their lab has been doing in Flywire.
Thanks, Mala. I'm Claire, and it's nice to see you all here. Thanks for coming. So Andrew is at the University of Puerto Rico and runs a lab with Steffi Hampel. And Sven, could you please play the recorded video?
Yes.
Okay, so the Seeds Hampel lab studies the neural circuits that produce grooming behavior, and we do this for several different reasons. For example, grooming serves as a great model for understanding how mechanical stimulations at specific locations on the body are transformed into precisely aimed responses. So on the left is an example of such a response from our previous work, where we painted iron dust onto the antenna and then used an electromagnetic field that's indicated by the light that pulls the antenna to elicit an aimed grooming response. So in that same work, we found that the mechanosensory Johnston's organ neurons in the second segment of the antenna are both necessary and sufficient for this response. In additional work, we and others have shown that stimulation of bristles that are located on the head and other body parts can elicit aimed grooming responses. So if you don't know this already, the neurons that detect bristle stimulations are called bristle mechanosensory neurons. On the right is a Galfor's organ neurons on the antenna, as well as bristle mechanosensory neurons that are located on different parts of the head. So one of our major goals in Flywire has been to define the neural circuits that produce head grooming behavior in response to mechanical stimulations. And our strategy for achieving this goal was to first identify the Johnston's organ neurons and the bristle mechanosensory neurons in the EM data set, and then we wanted to define all of their postsynaptic circuitry. So we started this work several years ago by reconstructing the Johnston's organ neurons in CatMade, and you can see that in the upper left. And we've since found and edited those same neurons in Flywire. We have also identified and edited nearly all of the bristle mechanosensory neurons coming from the head, and you can see those in the bottom left. On the right is a sampling of 200 neurons that we've identified as postsynaptic circuitry to these different head mechanosensory neurons. And we've also included a list of the different neuron types that we've edited in Flywire, just to give you an idea of the extent of our efforts. So because the mechanosensory neurons from the head all project into the ventral brain, the majority of the postsynaptic neurons that we've edited are found in the SEZ. And I'll just finish by mentioning that in the bottom right are the past and present members of our lab that have contributed edits in Flywire.
Thanks to Andy for sharing his lab's work. Sven, can you go on to the next slide? And so just one more example of a contribution from the community. This comes from Mareike Selcho. who has made an effort to identify octopaminergic neurons in Flywire. And these have contributions from many other labs, a lot of contributions, for example, from Greg Jeffress's lab for proofreading. But it's really valuable that Marijke has identified these and correlated them with their names from the literature. And this is an example of the kind of data that we're going to be making available in the spirit of openness. So now I'm going to hand off to Sven to talk more about the tools and the, well, other way around, more about data and proofreading and then tools.
Yes, thank you, Claire. So I'm going to start with looking back into the proofreading of Livewire, then projecting forward where we want to go and how we're going to do it, and finally talk about the tools that have already been developed by the community and that are ready to use. So Flywire has been around for about 2 years, and we are very excited that the community has been growing ever since. At this point, we have over 40 labs, and over 100 users have contributed over 500,000 edits. On the right, you can see where those edits have been made. So every point there is a edit in the brain, and the focus is clearly on the central brain. Another way to look at that is to use Nucleus Annotations. So in case you don't know yet, we have automatically detected nucleus locations. Those were created by Shangmu using a convolutional net. And using that method, he found about 140,000 nuclei in the brain. Now, those also contain also contain glia and also some false positive detections, but it gives us a good mark for how many neurons there actually are. We can now use these nuclei to also look at what has already been proofread or touched. So here on the right, I'm showing those neurons that have been absolutely not touched at all, which you can see are mostly in the optic lobes, the ones that have at least one edit, and a few of them that are outside of the segmentation. You may know that some of the segmentation in some regions does not cover the entire extent of the data set, so there are some nuclei that are outside. But overall, the vast majority of nuclei within the central brain has been touched and edited, and we also verified that further. So we believe, and Mala alluded to that this is now a point where we want to start an effort towards complementing this ongoing community effort in order to proofread the whole brain, and as a first step, the central brain. Having everything proofread to a certain extent is of benefit to everybody who is doing work in Flywire and also to the broader community. Now, the first milestone that we set out is the central brain. The reasons for that are that we already have possibly the majority of those neurons proofread, but also that they are of the bigger interest. Based on those nucleus detections, we believe that there are 40 to 50,000 nuclei in the central brain, though that needs refinement. Nucleus location goes only so far in determining that. So during proofreading, we will evaluate this further based on where the synapses of these neurons are. The second milestone, of course, then is the entire brain. So that is then the next step. The way how we are going about this already, because we actually started this drive some weeks ago with proofreaders at Princeton, is as a first step with a neuron-centric proofreading. So we use the nucleus detections to proofread all the neurons in the central brain first that have not been proofread yet. And we will track our effort there with the entire community effort by how many of those neurons basically make it into a list that indicate that they have been proofread. And then as a second step, we are planning to look at fragments that have a lot of synapses and attaching them. Those have probably been missed during proofreading, or those that have a nucleus outside the segmentation. And there we can track progress by looking how many of the synapses in a given brain region or neuropil are attached to a cell from step one. This effort is highly distributed, and we will also provide ways in NeurGlancer that anybody who wants to contribute, and we very much encourage that, of course, to basically pull a neuron to proofread and then submit such proofreading. The neuron-centric proofreading that at least people in Princeton are following is what we call a fast proofreading regimen. So we found that proofreading of single neurons can actually happen on average within 10 to 20 minutes. And we did some evaluation on that for the Flywire paper, where we compared this to very deliberate proofreading over multiple rounds and hours of proofreading, and found that very fast proofreading of the major errors is almost as accurate as the more deliberate proofreading. So again, yeah, we're looking forward to a distributed effort, and we are also planning to roll this out in a broad manner in about 4 weeks. And why 4 weeks, you will see in a minute. Because the way we track proofreading is bound to nuclei and neurons. So we're going to use those to track proofreading. And one example for that is the neurons that have been proofread at Princeton. So about 8,800 of these nuclei have been already proofread by proofreaders, and we're going to share that information with you. But in order to now continue proofreading and not redo work, we are also starting a annotation table, an effort to collect all that information that everybody probably at home has to make that available and share. So the plan is to have a centralized proofreading table that stores all the neurons that have been proofread and deemed, at least to some extent, complete by somebody in the community. Such an effort and such a table avoids duplicated effort by everybody, because everybody can check if a neuron has already been deemed proofread and does not need to go and check, which alone takes time, even if you don't make edits. It allows us to assign credit for the work that has been done for checking. And finally, it also enables more analyses, because everybody will have a bigger network that includes the neurons of their interest. So what we are suggesting and asking here is that the community basically builds this table, and we will consolidate all the tables that people are sending in. So over the next week, we are looking forward to collect proofread data. We will show in a second how this looks like, and then additional 2 weeks to consolidate it and make it available. All this data will be available through programmatic queries in Cave, but also a dedicated UI that does not require programmatic knowledge, programming knowledge. And also to give you an estimate of what you can expect there to be immediately is that just from the proofreading at Princeton and also at the Jeffries lab, we believe to have already over 25,000 neurons proofread, and so we will easily clear the 30,000 neuron mark, and that is information that is extremely valuable to everybody. As an example, this here is a table that, a Google Drive table that we have been working on in the Sanger-Murphy labs. And so we have been keeping track of coordinates, segment IDs, but also names, who identified it, and who said that something had been proofread. And what we are now collecting is essentially that kind of information. Coordinates are the best way to unambiguously say what neuron is actually meant with an annotation. Further, of course, if it has been proofread, a segment ID, if available, and also to assign credit credit who would actually assess the proofreading. Separately, we also want to start a table that collects cell names. Cell types are a very difficult and opinionated topic at times, but what we're trying to do is we're trying to collect a table of names that people assign to these neurons and make that available for queries so people can query this table without programmatically and non-programmatically to see if somebody that called a neuron with a certain name. So leaving that topic, one thing to mention in this realm is the principles of Livewire that govern how people interact with each other and how data is shared. Together with the advisory board, we discussed the principles and we made changes to the text of the principles, but we didn't change the principles. So the principles have been clarified to specify that assessment of a neuron is also credited. They clarify the expectation that somebody should have for the work that has been done. So 2 hours of work not leading to a big credit, whereas months of work are something that somebody should expect to be accordingly credited for. It also clarifies the expectation that we are all better off when we share the data instead of blocking each other from using it. So blockage of somebody using one's proofreading should be justified with an overlap in scientific questions, and there is a way, or there will be a way to basically raise a blockage to the advisory board, which will then try to mediate. And finally, we also will make better services available for people to query all this information in one place, such that you know who you have to talk to and who to assign credit to. Now, Flywire itself provides all the data and some tools to look at the data, and a lot of APIs that are programmatically available. And we are aware that this data can be used more effectively if we have more tools to interact with them. And on our end, we have obviously also limited amount of time to develop those tools, but we very much encourage everybody to develop tools and also try to facilitate them by supplying all the endpoints and APIs. There is a channel on Slack, the Annotation Infrastructure channel, which is also shared with the fancy community, which incidentally runs on the same infrastructure where this is discussed. And I encourage you to check that out. And there are maybe interesting things for something that you wanted to do already. Here in this town hall, we have introductions to 3 different kinds of tools that are available right now for everybody to use. The first one is Brain Circuits I/O by Stefan Gerhardt, which is a way to query the synapse tables and visualize it in Neuroglancer. We will hear from Philip Schlegel, who will present on Navis and NatVerse, developed by the Jeffries Lab. These are mainly programmatic tools, but then you will hear from Ari Matzlia, who developed Flywire Gateway, which is basically using Navis and makes some functionality available such that you don't need programming knowledge to use it. Okay, so with that, I will play a video for each of them. These are all 2 minutes long, and we will start with Stefan Gerhardt on Brain Circuits I/O.
Hello, this is Stefan Gerhardt. I'm an independent contractor working on the Brain Circuits I/O platform that has been funded in the past few months by Rachel Wilson and Wei Li. The platform offers you a number of apps that you can use for your proofreading and analysis workflows. In particular, I want to highlight the Neuron Connectivity app, which you can go to. It's freely available, and you can select the Flywire dataset in the drop-down menu here, and enter your segment ID for which you're interested in upstream and downstream partners. If I enter here partner and press add IDs, the ID is added here to the source table. As well, the main neurotransmitter type that is based on predictions of different neurotransmitters is highlighted as well. The upstream and downstream partners are visible in these two additional tables together with the synaptic counts. What you can do here is to select partners of interest and also downstream partners of interest. And you can go here to view selected, which brings you to the Flywire platform. And for those selected segments, you can see them here automatically populated. White is the source table and blue are the upstream and red the downstream segment IDs. So this is a very convenient way to find partners in the data set. There are also other tools which you can explore and that are useful for your exploration of neural circuits. Thank you for your attention.
Okay. Next up is Philip Schlegel.
Hi, my name is Philip Schlegel, and I'm a postdoc with Greg Jeffries at the Connectomics group in Cambridge. Over the last year or so, we have developed both R and Python tools to programmatically interact with Flywire and to bring data such as neuromorphologies into well-established neural analysis libraries. On the R side, there's FATBSEG that provides an interface between Flywire and the net ecosystem. On the Python side, we have FATBSEG-Py providing an interface with Navis. Although there are minor differences, mainly in syntax, most features exist in both packages. It's important to note that we heavily rely on tools and backend infrastructure developed and maintained by Sven, Forrest, Will, Eric, Casey, and others. In combination, these packages let you do things like interact with the upcoming annotation tables, fetch connectivity via the Booman et al. Synapse predictions, create and read Flywire URLs, query the Ekstein et al. Neurotransmitter predictions, fetch neurons from Flywire, transform neurons to other brain spaces, here the hemibrain, visualize. Moving on, you can also query the underlying segmentation, efficiently skeletonize neurons, or run NBLAST to, for example, create morphological clusters or find matches across datasets, and much more. If you have any questions, please find us on Slack or open an issue on GitHub.
And finally, we have Ari.
Hello, my name is Ari Matzliach, and I'm a research engineer in the Princeton Neuroscience Institute, working with Marty and Sung Labs. Today I would like to present two recently developed tools that you may find useful. The first one is Flywire Gateway, and it allows you to easily visualize any neuron from the fly circuit or hemibrain datasets in Flywire. For instance, if I navigate to the Fly Circuit tab, I can select the neuron and have it mapped to Flywire. After translating the coordinates and scale, I will get a direct link into Flywire where this neuron is visualized. Similarly, I can also upload any neuron as a SWC file and map it to Flywire from any of the supported brain templates. The second tool I would like to present is for visualizing connectomes, or connections of neurons. Here you can upload your data as a CSV file in the format specified here, and then have it visualized as a weighted graph. After selecting the layout and other visual options, you can download the graph as an SVG file and use it in your articles or presentations. I hope you find these tools useful, and please reach out with any feedback or questions. We will post the links in the Flywire forum Slack later today. Thanks and bye. Hello, my name is...
Okay, and after these great tools. I wanted to also give a very brief look at what is ahead from the Flywire side, what data and tools we are working on right now. Again, I also encourage you to check out our Slack channel where we have many of these conversations. So many of you might be aware that we are working now for quite a long time on an integration of annotation visualizations and queries such as the synapse with the nuclei in NeuroGlancer. This is still ongoing, but it's definitely getting closer. We have synapse-based neuropil information, meaning for each synapse in what neuropil it resides, that will be added soon to the synapse table for everybody to query and use. And then we are working on something that is called the L2 cache. In case you come across it, that will allow everybody to query skeletons for neurons right after they have been edited, but it also provides information like volume and all other kinds of information for neuron very quickly after an edit. And finally, based on skeletons, we're also working on integration with Neuron Bridge using color maps. So we had some very good conversations with the folks over there. Eventually, we basically want the Flywire community to be able to bring in a neuron from Flywire and query all Gulf lines or the hemibrain through Neuron Bridge, and vice versa by exporting first the original segmentation of Flywire to be able to query neurons in Flywire coming from the Gulf line or the hemibrain. Okay, and with that, I think we're coming to the end of the presentation part of the town hall. We of course want to acknowledge all the amazing people that helped make Flywire possible, and I'm turning it over, I guess, to Claire.
Thanks, Sven, and thank you to all the contributors. Now, feel free to post your questions in the chat and we'll have a discussion session. Please direct them to me, Claire McKellar, and I'll moderate. And I'll start off with one question that has come in. Sven, for a non-programmer, what are some of the tasks that you could do right now with the tools that are going to be shown in the forum later today?
The tools that I just presented?
Yeah, so it sounds like the question is, for someone who isn't a programmer, what are some of the first things that they can try doing?
I think the first things they can try doing is finding upstream and downstream partners for any neuron that they're interested in, and using Stefan's tool. If somebody has reconstructions in CatMade or the hemibrain, you can use the Flywire Gateway to find that same neuron in Flywire.
Great. Then another question that has come in is, do you guys intend to release a new segmentation?
Yeah, so I could take that. We have been working on realigning and segmenting the data, but at this point we don't have a timeline for that. And we actually think, given the status of proofreading of this data set, which has gone really well, that is sort of our best, route forward is to proofread and analyze this data set. So at the moment, no, while we're still working on it, and there's a potential for us to release a second segmentation, we don't have plans to do so yet. And I guess I would just add that, along with that, we would also develop methods to, potentially automatically port the proofreading over. So it isn't as if work would be lost, that, you've proofread circuits now, then there'll be a new segmentation and you have to start all over again. That's not our intention at all. If anything, it would just speed up the process. So we could port those proofread over, and then further proofreading would be accelerated. So I don't know if you want to add anything to that, Sven.
No, yeah, that sounds good.
Feel free to type your questions in the chat to me.
I wanted, while we're waiting, I just wanted to follow up on Sven's presentation. We are asking for lists of proofread neurons for all the reasons Sven mentioned. And so we're hoping that you all can provide those lists over the next 2 weeks. It's a bit of a rapid timeline, but we're hoping you're motivated by having access to all of them in the 2 weeks after that. So we're excited to get them. There'll be some work on our end to clean them up and organize them and make them available to everybody. But we'd like to do so as quickly as possible because we know how valuable having those annotations will be for everybody. And once that's done, as Sven mentioned, we can start this proofreading drive in roughly the same timeline, so in about 4 weeks. And we're really excited for everyone to contribute. As I said from the outset, it is a community project. People will get credit for proofreading. So even if it isn't just your circuit of interest, if you're just proofreading in the data set to contribute to the drive to get us to completing the connectome, you will get credit for that effort also. And so we're hoping that many individuals and many labs will participate in that drive.
Thanks. So another question regarding neuron naming and annotation. If we come across neurons we have functionally characterized and published on, but they're already proofread by someone else in the Flywire community, should we contact you or the people who proofread the neurons first?
So with regards to naming, it's okay for neurons to have multiple names. That is that the annotations in Flywire can handle multiple names. If you click on a segment, you'll see all names associated with it and the people who have identified and named those neurons. If the question is about use of neurons, did I understand the question correctly, Claire?
Well, who should be contacted first, Flywire or the people who proofread the neurons?
Oh, I see. Sorry, I don't think I understood. Contacted for what?
So if I'll just read the question again, if we come across neurons we've functionally characterized and published on, but they're already proofread by someone else, should we contact you or the people who proofread the neurons first? Yes, Sven, go ahead.
Yeah, I think for naming is independent of proofreading. These are two separate tasks, and they're both very valuable, and both are, according to principles, assigned credit. So I don't think that for that somebody needs to contact the person who proofread it, but rather, submit that information. And the first way would be through us, but our case not required for anybody to name a neuron, of course.
More questions? Okay, how can labs contribute to tool building? Are there a list of needed features that we could help with?
So we
didn't plant that question in the audience, but go ahead.
Yes. Yeah, we can create such a list. We had a discussion if we should have a list for this meeting, and but we will create such a list and we will post it.
Yeah, so we're trying to put our energy into the the infrastructure for Flywire as much as possible, because we know there's a lot of functionality that will help you all with proofreading, but then there's additional functionality in terms of circuit analysis that Drosophila neurobiologists really want. And so as many tools that you all are developing to do circuit analysis and are willing to share with the community, we welcome them. So any help we can provide on tool development, we are happy to. And we'll also make it easy for you to share those tools within the infrastructure of Flywire. So one of the tools we mentioned that Ari worked on is graph visualization. You can load up a list of neurons and see your connectivity as a network graph, and then manipulate that graph to display it in different ways. Those kinds of things can be really useful for circuit analysis. And at the moment, we have a limited bandwidth to develop things like that. So the more tools like that the community provides, I think will be really useful for everyone.
We have a contribution here regarding the previous discussion about naming neurons. So Someone posts, for the point about naming neurons that others have proofread, it would be nice to communicate with you, Claire, or Flywire, and the other group. Someone probably proofread those neurons for a reason, and early communication should be encouraged. That's a good point. Thank you. Then a next question. Thank you very much for all your efforts. Regarding proofreading, are there any important drawbacks to keep in mind? For example, how confident should we be with finer dendrites or axon terminals? In this fast proofreading method, can there be potential biases on proofreading difficult neurons like darker cytosol, narrower branching, etc.?
Claire, would you like to answer that question?
Sure, yeah. So the finer dendrites, so axon terminals luckily are pretty thick, so they're easy to assess. With skinny little twigs, even humans have a really hard time. And so that's part of why we are working on computational methods to try to attach orphan twigs, is because there are so many little twigs that it would be a really Herculean effort for humans to try to fix them all. And I frequently myself, I'm not confident when I'm looking at a twig about whether it should be attached or not. So we're going to try to deal with those as much as we can with future computational approaches. In terms of being confident with the quality, for example, in the fast proofreading method, there could be potential biases. Darker cytosol definitely makes it harder. Narrower branching definitely makes it harder. We're envisioning that a first release of a connectome would be kind of like a beta release, that we really see the connectome as sort of a living thing that continues to improve as everybody contributes to it. So we don't see it as a static thing that gets produced and published. If there are biases now, we can keep working on those. And if there are things missing now, like orphan twigs, we can keep working on those. And we hope that the whole thing will continue to improve over time. Thanks for that question. So here's one. How should people with similar interests collaborate? Mala, do you want to take that?
Yeah, no, that's a great question. So yeah, I was probably just going to say what you were going to say, Claire, which is that Claire has been trying to initiate some interest groups within the forum for people working on similar areas. In fact, when you sign up to use Flywire and you get trained with Claire, you provide her with some information on the circuits you're interested in working on, and based on that information, she can start to put people together who express an interest in similar neurons. Of course, your interest may change over time, and that's perfectly fine. And we encourage you to use the forum to communicate with other people about circuits you're working on and to find collaborators there. Another useful tool is looking at edits that have been made to neurons. So that's publicly available in Flywire. You can click on a neuron and see which users have made edits. If you need help identifying which labs those users came from, we're happy to do so. And that can help you. If you see that one particular lab has been making a lot of edits on neurons that are of interest to you, we encourage you to get in touch with them and collaborate rather than compete within Flywire. Our advisory board is more than willing to take up any issues that arise. So far, they've been pretty limited and easily resolved, but we anticipate going forward as we have more users and more of the connectome complete, that more, that issues can come up, and we're willing to step in to listen to them and resolve them. So you should reach out to us if you feel that something has happened within the community that's upsetting to you in any way, and we will work to resolve it.
And many of those situations could probably be avoided with early communication. So we really strongly encourage getting in touch with even someone that you might see as a competitor. Get in touch, because you can divide and conquer. You can decide, we'll do the left hemisphere, you do the right, or we'll do upstream neurons, you do downstream, or we'll publish back-to-back papers, or we'll all be on one paper together. There are ways of working together. And so we're hoping that this big push now to share neuron data and make all that information viewable by everyone will also just help prevent more overlap. And then also in the forum, you may have noticed that we're starting to create interest groups. So there's a non-neuronal cells interest group for our 3 labs who don't work on neurons. There's a SEZ interest group, and that one already has 40 members, which is really exciting. So you heard from Andrew talking earlier about their work on grooming, but also the SEZ is involved in several other behaviors, notably taste and feeding. So that I could imagine being quite an active interest group where the SEZ is not included in the hemibrain data set. So there's a really nice opportunity here where the SEZ community could pull together and do a proofreading drive there and probably get an SEZ connectome in just a matter of several weeks because of the number of people interested and how fast proofreading is going now. So I strongly encourage you to post your interest in the forum and keep an eye out for interest group pages that we're going to be creating to help direct proofreading efforts according to brain neuropil regions. Okay, another question. Will there be a system for evaluating and conveying the proofreading quality of a neuron? Proofreading from an expert who's been proofreading for a decade and proofreading from a beginner who have just begun may have different quality.
Yeah, that's absolutely right. So Claire mentioned that, the connectome will continue to evolve as we make more and more corrections. So in that sense, the work may never be finished as we, continue to fine tune the proofreading, but that's absolutely an excellent point that proofreading quality can vary. So, one of the tasks that we're asking for is, so there's, proofreading a neuron, identifying a neuron, but also assessing the quality of proofreading. So if, your lab has gone through and, said, oh, this neuron looks good enough, it's done, or this neuron needs more proofreading, that's also a task that is important and that we intend to assign credit for, is to do that assessment. And on our side, we're doing a bit of spot checking where we, are in some cases proofreading neurons a second or a third time in order to check the quality of proofreading, not of the whole data set, obviously. We're also setting up quantitative methods to evaluate the quality of proofreading throughout the data set, which we think will be really important to go along with any kind of connectome paper and release of the data set. So, those are things that are ongoing. But if you and your labs have assessed the quality of proofreading throughout the data set, please provide us with that information in a table, and that will be part of the commonly shared information.
So if I could just add to that regarding quality, what Mala was referring to about automated methods. We are creating a testing system for future users coming in so that we could give everybody the same set of neurons that we have ground truth for, that many experts have laid eyes on, and hold people to a certain threshold standard of accuracy before they would be allowed into Flywire. And one of the reasons that we are working on that system is not to make it harder for scientists to get in, but actually for a really exciting reason, which is that we hope to open up to non-scientists, including gamers. So the iWire game that was the predecessor to Flywire that was open to the public, we are hoping to recruit the top iWirers, people who often play so much that they're kind of working the equivalent of a 40-hour-a-week job tracing in iWire. So these are people who are pretty sophisticated with EM, but because they're not scientists, we do want to provide quality control before we allow them into the data set. But I have spoken to many of you, and everyone seems to be happy with this idea and excited about it, that all of our circuits are going to get finished faster by letting more people in. So you may see more features like gamification be developed in Flywire. More questions? I can also, anyone who would like to raise their hand, I'm happy to unmute people so that they can share their own thoughts if you don't want to type in the chat. But for now, I have one more coming in through chat. For sharing proofreading, naming, etc. tables that we've already generated, what's the best way to give those to you? Will there be an automated form or submitting, for submitting them? Sven, do you want to take that?
Yeah.
We wanted to set the threshold as low as possible, so we encourage you to send them as CSV or whatever is most convenient to you, as long as ideally with those columns as outlined. Moving forward in the future, we will have a tool to take on that information from a new answer state automatically, but that is more for a single neuron, not for a batch process.
And I'll send an email later today with all those columns that we're asking you for, so that you've got all that written in one place. Another question, would you like people proactively to share their ongoing work and future plans? If so, where? Absolutely. Sven is nodding vigorously. Sven, you want to?
I was nodding about the idea, and of course, sharing early and often, I think, helps everybody in the long run. I think what Claire said earlier about channels on Slack, I think that is the best way where everybody has access to and can see. That being said, everybody can create annotation tables that can be seen by everybody else. So you can have your own table where you outline with annotations for those neurons that you are working on, and that is also a way to share that information with everybody in the community.
A good point raised in the chat. I wonder if it would be useful to send a template for the submission of neurons. Great idea, thank you. I will do that when I send the email. I'll send out like just a little Excel sheet or a Google sheet or something. Okay, next question, 2 questions. I'll take the first one first. Is there a plan to unify the hemibrain, walled garden, and Flywire tracings and annotations? That could accelerate things massively. So let's just address that one first.
In one framework, no, but we realized the value of being able to make comparisons between hemibrain and Flywire. And so we mentioned a couple of tools developed in the Jeffress lab. And then we've built a little infrastructure, a little, user interface on top of that in order to make that easier. So visualizing skeletons within Flywire, for example, are going in the opposite direction. So at the moment, no, we don't have a plan to integrate hemibrain and Flywire FAFB data within one framework. I don't know if you want to add to that, Sven.
Yeah, what I want to add to that is that so when I discovered Neuron Bridge or was made aware of it, I thought this is a great way to integrate across multiple Fly resources, and so that's at least the motivation for me to work on that, to make that possible, to allow people to go back and forth between those data sets. So while this is not a single framework, it hopefully will make it way easier for everybody to go between different flight data sets, for going between CAP-made reconstructions and Flywire. I think that's because it's one data set that is the same data set, same fly. This is a little bit easier, and I think Ari's tool, built on top of the Jeffress Labs infrastructure, allows that quite nicely to put in. And we are also planning on using some of the published CAD-made skeletons to also verify quality of proofreading in Flywire, for instance.
Thanks. So next question, is there a plan to include the VNC in the tracing effort?
Yeah, I mean, we've had a lot of great discussions with Wei and with John Tuttle about integrating the data sets. They're collected from 2 different flies, of course, the female VNC, FANC, and FAFB Flywire. but they are both being edited in essentially Flywire infrastructure. So it's the infrastructure we built for proofreading that's being used for proofreading of Fancy. So there we see a path towards integration, and I think we should talk more about that. You know, there is work already being done at the interface of those data sets by Greg Jeffress and others, neurons that are in the brain and in the VNC. And so it seems like there may be a mechanism for that going forward. But at the moment, they're also sort of two separate communities working on these data sets. But we've been learning from each other. I think that they also have quite an open and active community for Fancy that's been it's been useful to have those discussions.
Great. Next, from folks that have done work on FAFB in CatMade and HemiBrain, is there any information that can be useful for you that you would like us to share? That would be great. There are, you know, those communities have their own principles in terms of service. So anything that you share from there has to follow their guidelines. But if you, well, for example, all the published neurons from the walled garden, we could use for helping proofreading in Flywire. That's one option. Sven, can you think of anything else that we can accept from people in those communities?
I think cell naming is definitely another topic that could be imported. The HemiBrain has been very good about that, or has put a lot of effort into cell naming. So I think being able to reference to the names that were given in the HemiBrain would be useful for everybody.
And actually, the next question had to do with names as well. Wondering whether names of neurons would be a subset of the annotations of neurons.
Names for neurons will be stored as annotations, yes. And did that answer the question, Claire?
Yeah, and I think, I can't remember if we mentioned it earlier, but we are going to allow multiple names. So I like to use the example of, say, giant fiber spelled ER or RE or GF or GFN. So neurons, of course, can have many names. And right now we're allowing them all. So everybody gets credit for contributing to identification, even if a neuron ends up with multiple names. And we may eventually come up with sort of a voting mechanism for people to settle upon a canonical name for a neuron. But for now, it's quite open. Next question, could the Flywire neuron name and annotation include HemiBrain ID for neurons that are unambiguously identical across the two data sets?
I think we can have a separate table that is just there to reference these two data sets with each other and do exactly that, match Flywire neurons to HemiBrain IDs. And that's
okay.
That will go on the list.
And I will take one last question. We're almost at time. Is there a canonical way to go from Flywire segment ID to potential split GAL4 halves? And if so, is there a set of directions somewhere for that?
You could field that, Sven. You were just mentioning the Neuron Bridge resource, so go ahead.
Yes, so the place that I would look is Neuron Bridge, and we have ways programmatically to create skeletons, and feel free to text me about it. You will also find information in that annotation infrastructure channel, and based on that, one can create color maps that one can take to Neuron Bridge to search, and that's a process that we want to make easy and faster. But I think it's already walkable at this point.
Yeah, with that, I'd like to thank everyone for coming. Feel free to get in touch with us if there were any questions that weren't answered here, and we'll be in touch soon with requesting these lists and providing more information in Flywire.
Thank you all for joining.
Yeah, thank you for coming.